Creating DNA profiles. DNA has been around since the beginning of time, but it wasn't until 1984 that Dr. Jeffries found a way to compare DNA samples. DNA profiling is used for comparing DNA samples from crime scenes to suspects to fraternity tests. Creating a DNA profile has many different techniques that are used for analyzing samples. The technique that a forensic scientist uses depends on the cost, the time, the amount, and the quality of the sample. Understanding the different techniques used for creating DNA profiles is important for someone in criminal justice. I will tell you about the different techniques that are used. The first method used for creating a DNA profile is RFLP, or Restricted Fragment Length Polymorphism. RFLP is hardly used for forensics today due to large samples amount needed for analysis. RFLP requires the examination of multiple sections of DNA strands to get the variation for comparison. This method is time-consuming and opens up the possibilities of human errors. The next technique is PCR, or polymerase chain reaction, and is usually the first method used in creating and comparing DNA samples. This method can replicate the smallest sample to get a larger sample for analyzing. This is done by adding heat-stable DNA polymerase to bind the DNA together, then heat to 200 degrees so the threads separate. Let the sample cool and repeat the process to get enough sample needed for an analysis. Next is the STRs, or short tandem repeats, which are very small alleles in the VNTR. Alleles are small pairs of genes that happen alternately at specific points, or LOCI, on the chromosomes. STRs are more accurate than other methods due to the sample size needed. AMP-FLP, or amplified fragments length polymorphism, is used to replicate DNA by the PCR process. It uses restricted enzymes. Then the fragments are amplified by PCR and finishes with sorting using the gel electromorphosis. This sample must be high quality or it will cause errors. AMP-FLP is frequently used because it is the cheapest and has automatic results. Once a DNA profile is created, law enforcement can use it as a reference sample against suspects. CODIS was created to house all these created DNA profiles so law enforcement can have a database to use for comparison. In conclusion, creating DNA profiles is very useful and helpful when needed to compare DNA samples. Creating these samples can now also be used for genealogy and for the testing of diseases. So depending on the cost, time variable, type, testing, and quality will determine the technique used when creating a DNA profile. Remember that everybody's DNA is the same, except for those 13 variables that stand alone and alter our DNA.